we're going to hear from Adam Resnick. Many of you may know him as he is the director of the Kids First Data Resource Center. And I will not take more of his time and pass the word to him. Adam.
Thanks so much, Marcia, and others. You know, for the sake of time, I'll actually be speaking on behalf of those who have done much of the work, and I really want to commend Yaron Guo and Jeremy Costanza, you know, from the Kids First DRC side for developing resources that really take sort of an orthogonal approach to some of what you heard already today from NARA, for example, supporting the queryability and findability of the resources investigators might want to access by leveraging gene-centric queries. And one of the first challenges that investigators often encounter, and we in the DRC continuously try to support and improve how we describe and define the resources within the program, is what data can I access right now or query, and what data do I have to approach dbGaP and requests. And this has been especially challenging for investigators as the program has grown, and we have, you know, tens and tens of different projects that each one of them has their own dbGaP requisition. And it's further complicated by the fact that there's differential access layers within Kids First, depending on the sensitivity of the data. So cancer data sets and their or other data sets that have expression counts or gene fusion calls are within a register tier that does not require access from dbGaP. And likewise, tumor normal studies don't require dbGaP provision access, but source data or variant data often does require dbGaP accessing. And it's that paradigm of complex layers of both access management and governance, aligning with NIH best practices and policies, plus the queryability of those resources for investigators that the DRC continues to try and improve and solve. So I wanted to just toggle back and forth between presentation, and I'll try and make it short, and the portal, just to give you sort of a real-time view of that process. You know, so investigators essentially, by accepting the terms and conditions of the portal, immediately enter the portal into the registered tier framework. And within this framework, the system already automatically knows and understands which data sets you already have authorization for within the portal from dbGaP. But as we've highlighted before, and I won't go through today, the portal does enable users to explore the cohort based on phenotypic and other variables, HPO terminologies, to create cross-disease cohorts. And for example, NARA and Hakon's cohorts are available for further exploration. That's one of the really exciting things about the Kids First program, is that data is released in very rapid order. So, you know, all the work that Hakon just described is based off the data that's currently available within Kids First for you and others to explore, both phenotypically and molecularly. Similarly, the brain tumor data sets that NARA described are also available within this portal, and can be, again, accessed, and likewise NARA's own EXO-1 cohort for exploration. The challenge investigators often face is, you know, how do I know which data sets to explore or utilize? And, you know, I just wanted to highlight, you know, the differences between cancer and non-cancer cohorts, you know, for some of that exploration. So here I just chose that same brain tumor cohort that Nara mentioned that she gained access to. And cancer data sets essentially permit users to explore the molecular data for the tumor normal setting. So here I'm just clicking at a single patient within that cohort, and we've empowered the PedsBio portal environment where users can gain row level data for this one patient and see all the derived mutational data for this particular patient. You can explore entire cohorts within PedsBio portal, and you can immediately query genes of interest like IDH1, for example, or IDH2 as Nara mentioned, and explore those mutational settings within the context of the assembled data. So here I chose, IDH1 and IDH2, and I can select these samples, and then I can actually review the specific mutations for that cohort within this setting. And the system will allow us to see a combination of the actual specific mutations across the genomic landscape of all of these tumors. Now, this study actually combines data sets across multiple data sets that we have been harmonizing, and I'll touch on that briefly a little bit later, really in alignment with Nara's comments. So here, for example, you can see, a cross-cancer cohort that includes IDH1 and IDH2 mutations. But the challenge that investigators often face really relate into the cohorts for variant discovery. And when we actually began doing the analysis on behalf of the DRC, it was clear that this was going to be a big challenge. And specifically, it's a challenge because there is a very large number of variants. And even actually, if you review simply the known pathogenic and annotated variants, there are still a lot of variants. So here is actually the known space of currently released data sets where we're just looking at the number of ClinVar annotated pathogenic variants across all the cohorts. Here at the beginning is Hawkins' cohort with the known annotated pathogenic variants. But known variants are typically not the challenge that investigators face. It's typically the unknown variants that are the challenge to phenotypically align. Let me just move my Zoom. So specifically, hold on. There we go. So specifically, we developed a variant explorer framework that allow users, even in the absence of dbGaP accession provisioning, to explore the context of the mutational setting for kids' first data sets. So here, for example, you can begin by looking at pathogenicity, and I'm just going to essentially highlight all the annotations, and I can already show you the data that came supported that chart that I just displayed, showing that there are more than 4,000 and already annotated pathogenic variable. And this table here essentially highlights the individual variants and the studies and the number of participants that are there. So for example, I can click on this particular number here, 3, and it'll essentially, what it'll do is it won't show you the row level data for that particular patients, but it'll show you the 3 studies that essentially contain that variant within them, allowing you as an investigator to further curate those studies phenotypically through the cohort builder, which we've demonstrated in the past, but then assess whether or not you would like to further request it. The other thing that I wanted to highlight briefly before the end is that many investigators already have information about a gene of interest. You heard this from Nara, for example. So Nara mentioned, for example, IDH1 or IDH2 mutations. So I'm just going to put IDH2 as a use case here. And immediately you can see a list of variants at IDH2. There are more than 2,600, but then you can further filter those variants and review them. So here, if I click on ClinVar, there's actually, a couple of variants that are already either likely pathogenic or pathogenic. A couple of instances of this in one particular study. And I can already see that this study is an orofacial cleft study that actually has germline IDH2 mutations that are pathogenic as identified. But the biggest challenge is the mutations that are not necessarily annotated. So those for which we don't have already verified pathogenicity. So for this purpose, let me just take one of the genes that Hawkins actually mentioned. So I'll start with I think this was POR. Yep. And part of the reason for showing you this is actually what I do as I watch presentations like Hawkins and Nara's and Joaquin's is dynamically actually query the genes of interest and assess them. So here, in order to review this particular gene, I can click on pathogenicity, and I can see that there actually aren't any already predefined ClinVar annotations, but I can begin to review predicted features. So here we have run a number of different prediction algorithms that essentially allow you to further annotate the gene. So here, let me show you, for example, combinations of queries that one can do. So here, I'm just going to click on, for example, high VEP annotations, or secondarily choose additional mutations as predicted. And then you can combine these types of annotation and say, I want either or of these predictions and or here. And then you can assess that leveraging these predictions, there are 26 variants that meet this criteria, and then you can further review the occurrences of those. And it is not surprising that Hawkins cohort is indeed here, and that there are 6 such variants that meet these criteria that potentially underpin the results that he showed. But the part that's really exciting, and I hope more of you participate in, is that you can see these predicted damaging variants also occur across a host of other studies spanning both cancer and structural birth defects. And it's in this manner that we hope we can support the expansion of the use of each one of these data sets and allow you to come at it from a gene-centric framework. Now, for time purposes, let me just highlight a couple of other efforts that are still nascent, but begin addressing some of the challenges that especially Nara highlighted. Joaquim already essentially mentioned, the interoperability that we're driving between Kids First and the Include DCC, and this will mature and allow users to essentially seamlessly interoperate between these data sets. But really exciting are efforts that are happening across the NIH, including the Common Fund Data Ecosystem and the NIH interoperability efforts that support the kinds of a nascent effort that Nara just described. So here we're leveraging an initiative by the NIH to do exactly what Nara described, where essentially, how can a user begin to assemble data sets from multiple locations? In this context here, we're leveraging the Common Fund Data Ecosystem portal and asking for specific features, again, spanning both Kids First data sets and actually GTEx data sets that we're working with. And I'm downloading a manifest of all those data. And that manifest then contains a list of all the files of interest, and leveraging new implementations within the NIH and across Cavatica and other platforms, I can essentially upload that data and intersect it with my data. Now, the uploading really doesn't move the data. What it really does is actually point to the data in their respective sources. So just like Nara mentioned, the data often resides across multiple endpoints across the NIH landscape. And how can you assemble it rapidly is a big challenge investigators face. And leveraging these new technologies that can assemble manifests that span across these resources across the NIH allow users to immediately bring all of these data assets into a common workspace, as long as they have the appropriate dbGaP permissioning and the system already knows that those exist. So here, essentially leveraging the system itself, I, as a user who has access to GTEx located outside of Kids First, can intersect it with data sets within Kids First. And this is some of the first efforts that we hope will address some of those pain points that Nara, you had mentioned. The other exciting advancement is partnership between Kids First and the NCI and Child Cancer Data Initiative with 2 upcoming data sets that are essentially dually positioned across CCDI and Kids First, one on brain tumors, the other on soft tissue sarcomas. And increasingly, we're seeing the use of Kids First data sets across these platforms, like I mentioned, with GTEx and or NCI and other format. And so you'll see Kids First located across NIH ecosystem frameworks, and hope we receive feedback from you on how to make it easier and better depending on how you enter in the system. Here, for example, I'm showing the Molecular Targets platform, a newly launched platform from the NCI, where they have leveraged essentially the neuroblastoma cohorts from Kids First and GTEx data, just like I showed you momentarily, to intersect, and you can assess the specific expression level of, for example, here GPC2 across this platform. Now we recognize that, as we try to address this complexity, we are also introducing complexity. We're introducing different resources, connecting across those resources to try and accommodate the entry point of the various stakeholders in this community. And we are continuously trying to improve and communicate about these resources and tools that allow users to connect data sets across these resources. In fact, Nara, the data set you're looking for exists within the NCI in ways that you should be able to bring into Chematica. And we hope that this will become easier and more routine as the ecosystem mature and as cloud resources develop. But we recognize it's going to be a continued partnership between you as researchers, us as DCCs, and the stakeholders that want to empower their data.